We started Nature League to explore the amazingness of life on Earth. Over the past year, we've explored a ton of different topics, formats, and themes, and had a lot of fun along the way. To celebrate that first year of content, we'd like to share a compilation video from our third month, which explored the theme of invertebrates. It began with a lesson plan about the names, relationships, forms, and awesomeness of this special group of species. When we think about life on Earth, it's easy to just think about vertebrates, or animals with vertebral columns. I mean, those are the animals we see most easily and most often, right? And they resemble us more than the creepy crawlies. But the thing is, invertebrates make up the majority of species in the animal kingdom. Let's check out this tree that shows the different groups of animals. See the chordate group? That's where vertebrates are. And that means everything else you see in the animal kingdom are invertebrates. So what is an invertebrate technically? Spoiler alert, it's not just insects. Invertebrates are technically defined as organisms without backbones, but what they are in practice is widely varied. Invertebrates live in almost every environment on our planet and can be many different sizes and shapes, everything from microscopic and spiny to massive and gelatinous. In fact, there are so many types of invertebrates that I think we should take a quick tour of their phylogenetic tree. A phylogenetic tree shows the evolutionary relationships between species and their common ancestors. So for animals, there was a common ancestor that gave way to the evolution of all other animals on Earth. And a phylogenetic tree can let us visualize the different lineages of species. Our first stop on the invertebrate tour, let's check out sponges. Sponges are in the phylum Porifera, and they're considered basal animals. This means their lineage is the closest to the original common ancestor. Because of this, sponges aren't too fancy. They lack true tissues and are sedentary, meaning they don't really move on their own. But don't count sponges out. They're super cool invertebrates in their own right and really useful in terms of human health. Sponges provide many compounds for human antibiotics, especially for penicillin-resistant strains of certain bacterial infections. And researchers are currently working on using sponge-derived compounds to fight cancer. Moving right along the tree, we find our next big split. This branch has to do with body symmetry, and the two groups are animals with radial symmetry and animals with bilateral symmetry. Radial symmetry is when you cut the body in several slices, like a pizza, and get something similar. But don't try this at home. Think jellyfish, for example. There are no clear left and right sides, just a top and bottom. Bilateral symmetry is when the body is symmetric on one plane. Basically, the body has a clear left and right side. Humans are a great familiar example of this. Let's check out some invertebrates with radial symmetry. These include the cnidarians and ctenophores. The silent C stands for cool, because who doesn't love jellyfish? The cnidarians include corals, jellies, and hydras, and ctenophores include comb jellies. Each of these groups have true tissues, but radial symmetry. So that leaves us with the rest of the invertebrates. They all have true tissues and bilateral symmetry, with a couple of exceptions. The next split in the tree is between protostomes and deuterostomes. But what they really mean is what happens in development. Protostomes have something called a blastopore turn into the mouth, and deuterostomes have that blastopore turn into the anus. You can always remember it because deuterostomes sounds like duty, which reminds me of butt. Now, besides having nice mnemonics for not forgetting those differences, what's interesting is that this split moves chordates like us and echinoderms like starfish away from the rest of the invertebrates. Kinda neat that some of our closest animal relatives look like this. So now that things with chordates and sea urchins have been sorted out, and we've left behind our jellies and sponges, we're still left with a crazy amount of invertebrates. There's a massive group called Lophotrochozoa, which includes a ton of smaller groups like flatworms, rotifers, bryozoans, brachiopods, segmented worms, and mollusks like clams, snails, squids, and octopuses. All of this, and we haven't even touched on insects yet. And don't you worry, we're totally going there next. Ecdysozoa are the last group we need to discuss, and they happen to be the most species-diverse group of animals on Earth. This group includes nematodes, or roundworms, and arthropods, which include insects, crustaceans, and arachnids. More than 1 million species of arthropods have been described by scientists so far, and these are just the ones we know about. The majority of arthropods are insects. In fact, insects are more species-rich than all other forms of life combined. The majority of insects are winged insects, which include beetles, flies, mosquitoes, ants, bees, wasps, butterflies, moths, true bugs, grasshoppers, crickets, and others. If you're overwhelmed by all of the groups of species we've covered in the last few minutes, it's kind kinda the point. Invertebrates make up the overwhelming majority of animal species on Earth, and we may as well give them some respect. Speaking of respect, invertebrates make up a gigantic amount of biomass, and that means they significantly affect the landscape, water, atmosphere, and other life forms here on Earth. They consume significant amounts of plant matter, other animals, and cycle nutrients by decomposing materials. They serve as food for larger organisms and even make up the majority of protein in some human diets. And just think, invertebrates pollinate crops, which is fundamental to our well-being. Now, in this lesson plan, we've only scratched the surface of the complex relationships and diversity of invertebrate species. Phylogenetic trees are constantly changing based on new information, so some of the branches on the tree we've discussed here very well may change. One thing is clear though, when it comes to the animal kingdom, invertebrates are where it's at. Invertebrates are found almost everywhere on Earth, and that includes here in Montana where we film. In this month's field trip, we visited the University of Montana Biological Station at Flathead Lake to learn about a notorious invertebrate within these waters. In terms of surface area, Flathead Lake is the largest U.S. freshwater lake west of the Mississippi River. It has its own stunning beauty and many stories to tell. I decided to visit Flathead Lake for our Invertebrate Month here on Nature League because one of these incredible stories involves an invertebrate, the mysis shrimp. Flathead Lake was a chunk of ice until about 15 to 20,000 years ago. In the late 1800s, accounts of Flathead Lake detailed about 10 species of fish. So at this point, fisheries managers saw an opportunity to stock Flathead Lake with a ton of fish that the public would enjoy fishing and eating. State and federal managers intentionally introduced about 25 fish species into the lake. One of these was the kokanee salmon, and people absolutely loved fishing and eating them. That meant major money for the local economy, so fishery managers were like, we should make sure that the kokanee have plenty of food. They introduced what they thought would be a better food, the larger freshwater shrimp mysis. What happened over the next few years has become one of the most famous examples of a trophic cascade ever recorded. Between 1984 and 1989, kokanee and Flathead Lake went from over 300,000 individuals to 0. See, the mysis shrimp eat zooplankton, which are also invertebrates, but much smaller than the mysis. These zooplankton were the original food for the kokanee, so the mysis started directly competing with the kokanee for food. The kokanee couldn't even eat the mysis because mysis hang out on the lake bottom during the day while kokanee feed near the surface. However, the introduced lake trout and lake whitefish feed on the bottom. Hello, mysis shrimp. Instead of the preferred kokanee salmon eating the mysis, the introduced lake trout and whitefish had an everyday buffet. Their population skyrocketed while kokanee disappeared. Fast forward to today, and Flathead Lake is dominated by 3 species introduced by humans: lake trout, lake whitefish, and mysis shrimp. But the coolest part, in my opinion, is that researchers at the Flathead Lake Biological Station were able to monitor and study this entire process and are still actively monitoring mysis shrimp and other invertebrates today. For this field trip, we joined a crew for late-night invertebrate sampling on Flathead Lake. But first, a word. Not from our sponsors, but from the dictionary. Welcome to this month's Wild Word. Once a month on Nature League, we'll look at the etymology, or origin and history, of words related to nature. This month's theme is invertebrates, so we're going to examine a word that is used when talking about the most speciose group of invertebrates. The word is insect. Like our other wild words on Nature League, the word insect is a pretty common word. However, a true breakdown of the word helps inform us about the group of animals it describes. You may have heard before that the sect part of insect derives from a root word meaning 6. And that seems to make sense because we think of insects as things with 6 legs. However, this isn't exactly correct. The word insect can actually be traced back to something a little bit different. Insect comes to us from Latin and can be split into 2 parts. The prefix in is a Latin word meaning into. So far, so good. But what about the sect part of the word insect? It's actually from a noun form of the Latin verb sectare, which means to cut. This is really straightforward word-wise, but in my opinion, the elegance is in how perfectly this denotation fits these organisms. I mean, just look at them. As biologists long ago started using this term to describe this group of organisms, they were definitely recognizing that this was a unique body plan, unlike anything else on Earth. So while a formal definition of insect is any of a class of arthropods with well-defined head, thorax, and abdomen, only 3 pairs of legs, and typically 1 or 2 pairs of wings, insect literally means to cut into. Nothing like being named after what you look like, right? Though insects do in fact have many different defining traits, it's undeniable that they look cut into. Literally, they look insected. And that is pretty wild. The mysis shrimp do a vertical migration daily when they feed, so we had to wait until it was pitch dark for them to come up from the bottom. This meant timing our trip for the new moon and waiting to sample until late that night. We sampled at 3 different depths using a tow net. We used a red light, which is invisible to the mysis shrimp, but let us see what we were doing. At each depth, we took 3 different tow samples and filtered them down into falcon tubes to take back to the lab. The next morning, we sat down with Jim Elser to chat about the sampling we did, as well as the biostation itself.
A biological station is essentially like a super collider is for physicists. It's a facility for research discovery and learning. In the wilds often, sometimes in the fringe lands, sometimes in farmlands, but it's where biological, ecological processes take place, and you can study organisms, ecosystems more easily, and experiment in them in ways you just can't possibly do when you're, on campus or in a city.
With mysis, even though that's not like your personal project, that is a definite personal project for Flathead Lake. And so what's the sitch on mysis?
Mysis is when it's an adult about that big, we saw them last night in our samples, it's a malacostracan. It's related to lobsters and crabs and krill. If you look at it, it looks like a krill. Common name is opossum shrimp. The biological station was here. It's been here since 1899, and the scientists were sampling the lake. They started to sample for mysis before they arrived, actually. It just goes to show how important it is to monitor ecosystems, especially valuable ones like this one. So our scientists sample this lake for all kinds of things. Year-round, around a very regular basis, using very standard methods so we can see what's happening.
Right, and monitoring, again, in situ, on site, is one of the coolest things that you get from having something like a biological station. So
that's right.
We should totally check out all the little guys that we found last night.
Absolutely, I think it's going to be fun.
Then we got to check out some of our water samples under the microscope. We saw all kinds of zooplankton, as well as mysis. It's no wonder fisheries managers thought mysis would be better food for the kokanee. These shrimp are massive compared to the other zooplankton.
Opossum shrimp, those are the eyes. And these ones with the short antennae are cyclopoid copepods. They're another crustacean. The tiny little filaments that we see are the diatoms. Another thing you can notice while they're swimming is how jelly-like everything is when they go past. It's because it's a small spatial scale where the viscous properties of water dominate. And so these things are like living in a swimming pool of
hydrogen bonding, like.
Well, no, it's like you were swimming in a swimming pool of molasses.
Oh, sure.
It's similar to if you were in molasses, what they experience.
The story of mysis shrimp affecting an entire ecosystem is just one example of how tiny invertebrates can have massive effects. Places like the Flathead Lake Biological Station are monitoring these effects and discovering new things all the time. Flathead Lake is an amazing place to visit and ridiculously beautiful on the surface. But what's even cooler are all of the stories unfolding underneath. Lakes aren't the only place where invertebrate drama unfolds. There are also some incredible processes in the ocean when it comes to this group of organisms. We next explored invertebrates in a format called Denatured, where I broke down a popular peer-reviewed journal article on the effects of ocean mixing on invertebrate species. For this month's Denatured segment, we're going to look at an article released in April 2018 in the journal Nature. In this month's lesson plan, we talked about the different types of invertebrates here on Earth and some of the ways that they significantly affect the planet. I mean, there are just so many of them, and scientists are only beginning to uncover their profound impacts. This paper, entitled Vertically Migrating Swimmers Generate Aggregation Scale Eddies in a Stratified Column, investigated the very large effects that these very tiny organisms might have on mixing the Earth's oceans. So here's what's already known. There are several examples of organisms affecting the flows of materials around them. For example, super dense groups of bacteria can generate convection patterns, and individual jellyfish can create significant water drift just by swimming. A lot of work on this so-called biologically generated turbulence has been done with marine zooplankton. Marine zooplankton are typically microscopic animals that either don't move or are too small to fight against the current. This means that marine zooplankton are typically tiny drifters. However, some are bigger and some are able to move more than others. Like everything else about life, exceptions abound. Previous work has shown that centimeter-scale zooplankton can form dense bunches that stretch out vertically over tens of meters as they migrate hundreds of meters up and down during the day while they search for food. To put that in perspective, that would be like 1,000 people standing one on top of another, moving as one blob up and down a distance of almost 17 kilometers during the day. So it's pretty easy to see how these diurnal, or daytime vertical movements of zooplankton might have some big effects on the water they move through. And in oceans, all of that water can have biologically important bits inside. things like oxygen, carbon, and nutrients. And even though each individual zooplankton is small, the vertical height of zooplankton swarms is not. And scientists have documented large-scale effects on carbon and oxygen distribution from these vertical swarms. So, although large-scale effects have been seen, it turns out that directly observing the fluid dynamics surrounding the zooplankton swarms is hard to do, especially since scientists can't easily predict when and where the migration events might happen. To address this phenomena, the research team set up experiments in the lab using the centimeter-scale swimmer Artemia salina. A salina is a type of brine shrimp, and if you ever had sea monkeys as a child, you've seen hybrid relatives of A salina with your own 2 eyes. Sorry to spoil the sea monkey magic, but sea shrimp might have been a bit closer to the truth of your childhood aquarium. The scientists set up 2 separate tank facilities for 2 separate goals. The first tank was 1.2 meters tall and was used to measure irreversible mixing. And the second was 2 meters tall and used to try out several water flow visualization techniques. So let's figure out what the team was actually trying to measure. Irreversible mixing in this situation is having a different density profile in the tank at the end of the experiment compared to the beginning setup. The irreversible bit means that energetically it is very unlikely for the system to return to its starting condition. Alright, so the researchers want to measure irreversible mixing due to the movement of the shrimp. That means they had to get these guys moving. In order to get the shrimp to move, the researchers took advantage of the fact that A salina is phototactic. That is, it moves in response to light. The 1.2 meter tank used focused LEDs with blue filters to create a vertical column of light. The 2 meter tank used a blue laser to create a column of light that was half the diameter of the other tank. Using light to coordinate vertical migrations inside the tanks allowed the researchers to simulate how zooplankton migrate in a non-lab environment. To measure irreversible mixing, the researchers set up 2 stable layers of different salinity, or salt concentration, inside one of the tanks. Once the water column was layered, the shrimp were introduced and allowed to adjust to the environment. To start the experiment, the researchers used a green LED light at the bottom of the tank to get the shrimp to gather there. Then it was time to migrate. They turned off the green LED at the bottom and turned on the blue LED light at the top. The animals began to migrate upward with speeds reaching 1 centimeter per second. Not exactly speedy for us, but definitely a good pace when 1 centimeter is your entire body length. Think about traveling the distance of your height once every second. It's not exactly lightning speed, but it's certainly faster than leisure walking. After 10 minutes passed, the top blue light was turned off and the bottom green light was turned on, so the shrimp would go back to the bottom of the tank. After 10 minutes at the bottom, the lights switched again. The cycle was repeated 6 times for a total experiment of 120 minutes, and the researchers got density profiles of the tanks each time. In general, the researchers were looking to find out how much mixing these little guys could do, and also how quickly. So what did they find? Overall, the team definitely found that irreversible mixing had occurred. More impressively, the shrimp were able to mix the water 3 orders of magnitude more efficiently than salt just diffusing through water on its own. That's about 1,000 times faster. Visualizing the flow of materials inside of the water helped the scientists figure out the actual process of the irreversible mixing they observed. They found a movement signature that might explain how the mixing happens. As the shrimp move upward, they actually push the surrounding water downward. And when there's a swarm of the shrimp, the collective downward jet of water is significant. The scientists also noticed whirlpool-like motions in the water right outside of the collective migration, and that eddy motion contributed to the mixing as well. The scientists concluded that the kind of mixing and downward jets of water observed in their lab experiments could create large-scale vertical motion and mixing of water in the ocean. Moreover, the fact that many ocean invertebrates engage in this kind of vertical migration means that their movement could be a major player in the motion of the ocean. This paper was published in the journal Nature, which is one of the most prestigious science journals around. Here are some reasons why I think this study wound up in this edition of Nature. First of all, scientists in previous research have estimated that the migrations of ocean zooplankton might affect ocean mixing just as much as wind and tides, and that is a major deal. Ocean mixing affects all kinds of important things, including the spread of heat, carbon dioxide, nutrients, and even the circulation of the ocean, which, by the way, is a big deal for life on Earth. In general, I think the main reason this study made the cut in terms of interest and importance is because it's of great relevance to climate change. If zooplankton have this big of an effect on the movement of the ocean, researchers who create climate models can't leave the little guys out of their calculations. And this is especially important when it comes to modeling the storage of carbon, because the ocean is one of the biggest carbon sinks on Earth. As our climate continues to change, change, potentially in detrimental ways, being able to model these changes is of great interest to scientists and the public alike. In terms of critiques, the biggest issue with lab experiments is that they are exactly that, lab experiments. These results weren't obtained from studying zooplankton in the ocean, so it's hard to know if they apply to the real situation. The first thing of note is that the species of brine shrimp they used, Asalina, doesn't actually live in the ocean. However, Asalina is similar to the marine zooplankton of interest, and the scientists made sure that the density of animals they used in the setup was comparable to the abundance usually seen in the ocean. There's also the issue of time and scale. Did the experimental setup really mimic what would be observed in the ocean? The duration of the experiment did match up with the estimated time it takes for an oceanic daytime vertical migration. However, a water column in the ocean would be disrupted by multiple individuals over the 2-hour period, whereas in the lab experiment, the column of water was affected by the same individuals multiple times. Overall, it's hard to say if the results seen in this study will perfectly apply to ocean migrations of zooplankton. However, the scientists mentioned that they already have plans to use acoustic equipment to measure these mixture signatures in the ocean, meaning we might see an in situ or on-site result at a later date. So here's the thing, on a personal note, I sort of just love the fact that these tiny organisms can have such a massive impact. We know that invertebrates make up an insane amount of life on Earth, but there's still so much we don't know about how they affect our world. And this study is one example of how scientists are figuring it out one experiment at a time. To round out the topic of invertebrates, I enlisted the help of my good friend Adrian Adams. In our third installment of From A to B, Adrian asked me why certain invertebrate species are cuter than others. So, Adrian, what do you have for me?
Okay, so why is it that millipedes are totes adorbs, and yet centipedes are essentially hellspawn? Why is it that when I see a millipede, like, scuttling along, because that's the sound they make, why is it when I see a millipede, I'm like, oh, look at the baby? And when I see a centipede, my first thought is, oh, kill it with fire. Because it's a deep internal thing, you know? I see centipedes and like earwigs and things like that, and there's something like deep inside me that says, nope.
Why the nope?
Yeah, why the nope?
Okay, so that gives us two different things to touch on, which is what are some basic differences between millipedes and centipedes, and then also probably need to talk about what makes something cute, right? What makes you go, the baby.
Look at the baby.
Look at the baby. Versus kill it with fire.
Yes.
Right? So maybe let's hit the first one first, and maybe the information we learn along the way will inform us about the baby versus blah, right?
Sure.
Good. First big difference, which probably goes right along with being cute versus being terrified. Centipedes actually are venomous.
I have seen plenty of videos where centipedes are like, you know, you've got your very posh nymph swimming to Attenborough, and it's like, oh, and the centipede has found its prey, and it's like some cute little mouse or something, and it like hisses. I swear to God, I've heard a centipede hiss. Shouldn't be possible. Maybe they just add it in post. It like kills these things. It just kills them. It's horrible.
Well, so that's another thing, is that centipedes and millipedes actually have different diets. So centipedes are carnivorous, so they eat other living things, like, so they eat other animals, right? Or things that are not plants, so things in non-plant category, right? Whereas millipedes are much more like, they'll kind of be detritivores, like eating things on the forest floor, or plant materials, like they're more scavengers, right?
Sure. Who are they hurting? They're not hurting nobody.
Yeah, so there's a difference, I think, in cute versus kill it when it comes down to something being carnivorous or a scavenger, right? Like that kind of informs us about something we feel inside that's different when something is actively hunting versus like, I'm gonna grab some leaf litter.
Leaf litter?
The role in the food chain, I think, for humans informs a little bit of that internal, like, versus, oh.
Well, a centipede could hurt us.
Also that, yes, it's totally fair to be afraid of something because it is venomous. Although the kind of venom we're talking about wouldn't necessarily kill a full-grown human,
Would
right?
it hurt?
Most likely, yes.
That would also explain why centipedes, at least from what I've seen, are usually like red or yellow or orange.
Which is a mixture of red and yellow. The more you know.
Da-na-na-na. We already recognize that internally as poisonous.
Yeah.
Or venomous. Ugh, I feel like that's a whole other topic.
Sure is.
Oh God, no! Abort!
Abort.
Okay.
So there are those. So if you look at their heads, so the actual body plan now is another one of these big differences. It's these modified legs at the very first.
Those are legs?
The very first body segment.
Those are legs? I thought those were pincers.
Right, but they're modified, right? It's the same, like if you think about the genetic blueprint there for each of the body segments is like a leg, right? So this appendage.
Is that what it is?
I'm pretty sure it's a super like modified leg that has
No,
venom on the ends.
like one leg per segment?
Yes.
On a centipede?
So centipedes have one leg per segment, whereas millipedes have 2 legs per segment. And I mean that on just one side of the body. So,
Yeah.
So per segment, a centipede has 1 and 1.
Uh-huh.
For a millipede, each segment has 2 and 2.
Okay.
Which also has a little bit to do with how they move, which I think is a piece of cuteness. So something that has 2 on each segment has a little bit more of this, right?
It's got their legs
Right.
totally little cross as they scuttlebutt.
And something about scuttlebutt is just cute.
Oh my God, scuttlebutt.
There's just, right? There's something different about scuttling versus like crawling.
And as we were talking about their head structure, the reason I made this disgusting face
Yeah, I saw that. I ignored it.
is because I was thinking about the millipede head. It's just this rounded, smooth, roundy little boop-boop-a-doop.
Yeah, and smoothie roundy boop boop-a-doops, fascinating enough, there's something cute about roundness,
Yes.
right? And I think that comes down probably to this evolutionary feeling of like sharp is harmful, is pointy, right? You're talking about things that are red or yellow. Also the idea of sharpness.
Centipedes look very rigid. Their legs look.
Right. There's a sharper feel.
Yeah.
And their actual bodies for centipedes are more flat, whereas centipedes are actually more rounded. And there's something with like what we like about general features. There's something about roundedness that psychologically for humans, we read it as cute. Probably because it can't hurt us or usually doesn't.
Okay.
Like rounded things, there's just that kind of human baby likeness of round. Look at the baby! My God, we figured it out.
We've come full circle. Circle.
Rounded,
Roundness.
cute baby. Look at the baby.
Ah!
Yeah, in the murder version of this, there's a bunch of red lines and yarn, and it leaves.
Okay, now what you don't know is.
Exactly. Yeah.
Yeah.
The conspiracy theory version. So you're exactly right. The little heads even are more rounded, whereas for centipede, you have, of course, we have the pincers, these modified pieces out of the segment, which have venom. So I think that there are definitely some very real, very physical characteristics that make them different.
Okay.
All right, but in terms of what that makes you feel and like whether you find something cute or find something deeply bothersome.
Probably goes back to childhood for me. Little pill bugs, little potato bugs. You know, everybody knows little potato bugs, little isopods.
Isopods, very good.
Oh yeah.
Proud of this boy.
Yeah. Little isopods used to be my favorite thing in the world. You know, you could poke them and they were soft and you could just pet them and they'd roll up and you blow them off your hand and back into the leaves. But whenever you see like a centipede, you lift up a rock and there'd be, a centipede or an earwig and just this, you know what I'm talking about, this.
It's a new method of teaching called yelling and doing this.
Well, when you're younger, you go a little bit more off, there's, I think, a little bit more instinct because you don't have so much book learnedness. So your brain just kind of functions on, I should stay away from this thing.
Yeah.
And you were saying before that the venom wouldn't be able to take down a full-grown person. So me as a child being afraid of that, oh my Lord.
Yeah, it's true. And things killing our children is actually way worse than killing us. I think there's also another piece at play, though, which would probably be media representation, right?
Because there's no human millipede.
And if there were, probably be rounded and cute. Even our pop culture has understood that centipede was the way to go.
Yeah, I guess. My cheeks hurt so bad. Oh God, that's good.
Yeah. And was there a video? What was the video game? Was it Millipedes? It was Centipede, wasn't it? I'm pretty sure it's Centipede.
Yeah.
And that would make sense because it, this kind of like video game, like evil, you're trying to avoid, or it's causing destruction.
Have you ever played a video game in your life?
No, I'm really trying hard here.
Okay.
Relating to people is hard.
So in pop culture, we there's kind of a collective whenever centipedes, are involved.
Yeah, which we have discovered in this conversation, probably for a reason and not just blatant discrimination, but based on things about them and what they do and what role they serve in the ecosystem and how they look and the fact that they're venomous, right? All this stuff. Those things combined are probably why
Millipedes are totes adorbs.
I think both centipedes and millipedes are totes adorbs, but I am totally respectful of those who find centipedes scary or yucky because of those things.
Our third month was all about invertebrates, and we had a ton of fun researching, creating, and filming the content you've seen in this compilation. Thanks so much for watching, and if you'd like to keep going on Life on Earth adventures with us here on Nature League, make sure to go to youtube.com/natureleague, subscribe, and share.